The biological effects of DD water. Deuterium. In nature, three isotopes of hydrogen occur. Hydrogen H mass number 1, deuterium D mass number 2, and tritium T mass number 3. Deuterium is the stable, non-radiant isotope of hydrogen. It has been known for decades that, due to the mass difference between H and D, molecules with a D content behave differently in chemical reactions. 1. 2. The numbers refer to those of the bibliography at the end of the chapter. For example, if a chemical bond contains deuterium instead of hydrogen, during the chemical reaction, this bond splits approximately 6 to 10 times more slowly. A hydrogen is replaced by deuterium not in the splitting bond but at a more distant point of the molecule. Chemical reactions also slow down to a significant degree. The first method of heavy water DEW production was based on the observation that, during the electrolysis of water, the decay rate of HW can be seen as a multiple of that of DEW. These so-called kinetic isotope effects allow a special insight into the mechanisms of chemical reactions, and the replacement of hydrogen with deuterium is widely applied in chemical research. Nucleomagnetic resonance experiments clearly reinforce that the presence of deuterium also has an effect on distant points of a given molecule, greatly influencing the behavior in chemical reactions of the molecule 3. The chemical difference between deuterium and hydrogen is manifested in biological systems as well. In recent decades, heavy water has been used in great concentrations during experiments, and it was observed that this significantly influenced processes occurring in the given biological system. 4, 5, and 6. In these experiments, it was stated that, for example, the growth of tobacco plants slowed dramatically due to increased DEW concentration. A dramatic effect was observed in the case of the mold fungus Aspergillus niger. This, as indicated by its name, is a black-colored fungus, which, in a heavy water medium turns alabaster white, which means that the fungus is unable to produce the pigment responsible for that black color. Drinking DEW worsened the overall blood count in animals, or, in the extreme case of drinking heavy water of more than 35% deconcentration, the animal dog perished. Research carried out with mice and rats showed that in mammals, as opposed to simpler living beings, hydrogen cannot completely be substituted by deuterium. Animals endure an approximately 25% of body fluid, which can be reached by drinking heavy water of a 30% deuterium content. The effect on living organisms of heavy water is not surprising if we take into consideration that a significant part of living organisms is made up of water, and that heavy water differs from normal water HW in many aspects too. Its melting point is almost 4 C, and boiling point 1.5 C higher. Its density is 10% higher, and viscosity 25% more than that of natural water. All these support the generally accepted view that the structure of heavy water is stronger than that of normal water. Part of the hydrogen present in living organisms, H atoms bound to oxygen, sulfur, and nitrogen in a heavy water medium, is quickly subtitled by deuterium. In this way, hydrogen bonds responsible for the stability of proteins become deuterium bonds. But deuterium bonds are stronger than hydrogen bonds. This, in turn, explains why proteins in heavy water are more stable when exposed to denaturalization and conformational changes. 5. A common feature of experiments referring to the effect of deuterium on living organisms carried out up to now is that they did not assign any significance to naturally occurring deuterium. Heavy water, therefore, was applied primarily in great conventions. In what concentrations does deuterium occur in nature? The D content of living organisms is defined basically by the D content of ocean waters and the evaporating precipitation in the form of rain and snow. Measuring the range of D content at several hundred points of the globe, It can generally be stated that the D content of precipitation decreases when nearing the North and South Poles and from the oceans towards the continental inland, as well as in proportion to height above sea level 7. This observation can be explained by the difference in steam pressure between HO and DO or HDO2. In our climatic zone, the D content of surface waters is about 150 ppm with minimal fluctuation as opposed to values of 155 ppm in the equatorial zone and of 135-140 ppm in the northern part of inland Canada. The 150 ppm value of D concentration is expressed in terms of mmol concentration. We find that in natural water, the concentration of H2 is 8.4 ml. This, as deuterium is present in natural water mainly in the form of HDO, in fact means a 16.8 ml HDO concentration. Taking into consideration that 60% of an adult's body is made up of water, and also considering that organic compounds other than water also contain deuterium, we determine that the D concentration of our body is somewhere between 12 and 14 ml. Quantitative ratio of deuterium in the human body can be demonstrated by the following figures. In a 50 kilograms person, there are approximately 5 kilograms hydrogen and 1.5 grams deuterium. As a contrast, human blood contains approximately 2 ml calcium, 1 ml magnesium, and 1 ml potassium. Taking the above levels into consideration, the question as to what role deuterium plays in biological systems becomes obvious. The presence of certain elements, calcium, magnesium, potassium, which occur in much lower concentrations in human blood and within a narrow range of concentration, are indispensable for biological functioning. Living organisms can differentiate between D and H beyond physical fractionating. It has also been known for 20 years that in different biological systems and within them, in certain molecules, the DH ratio may significantly change in contrast to the DH ratio of surrounding waters, depending on, for example, whether the plant fixes carbon dioxide from the air via C3 or C4 pathways. The extent of the decrease in the concentration may differ, or, in plants belonging to the so-called CAM group, in certain circumstances de-enrichment concentration occurs 8. This means that by defining the D-concentration of a plant, we can tell which of the above groups it belongs to regarding photosynthesis.